So the Golgi apparatus, you might see it also called Golgi bodies or Golgi complex. It just depends what book you look at. It's the same thing. It's just different names for the same thing. When I taught freshman bio, I used to tell them it was to remember it as stacks of sacs, because it's actually a stack of flattened sacs. And so when you look at the endoplasmic reticulum, it's sort of long and skinny, and then when you look at the Golgi bodies, it looks more like this, like 3 or 4 little short stacks, or little short sacs, I should say, in a stack. So that's what the Golgi bodies or Golgi apparatus looks like. Its job is to modify the things that the ER makes. It also makes a few things on its own, but I would say if you were going to try to remember sort of a key about it would be this, that it's sorting and packing. It's sort of like the UPS of the cell. It's a pack and ship. It's going to pack everything up for its final modifications, and anything that's going to get shipped out of the cell, that's going to happen here. And that stuff is going to leave the Golgi bodies in what are called transport vesicles. I mentioned yesterday that all of these cell organelles sort of form what's called the endomembrane system. So when something leaves the rough ER, for example, and it's going to go to the Golgi bodies, and there's a diagram of this coming up, this product would be in a vesicle. When it went to the Golgi bodies, that vesicle would literally fuse with and become part of the Golgi bodies. And then when that product gets modified and then goes to leave the cell, another vesicle would break off, and that would sort of fuse with and become part of the cell membrane. So pieces of membrane are literally breaking off and getting attached to new locations back and forth. So membranes are sort of getting moved around, and that's why the whole thing is called the endomembrane system. This next little thing I have in red, you won't be tested on. It does have 2 sides. The cis side is the side where the stuff comes in, and then it modifies it and leaves from the opposite side called the trans side. But you can put a note, if you want to, that you would not be tested on that information. You don't have to know the specific details of that. In general, from a picture, it's a stack of sacs, and we'll do some practice with identifying the parts from pictures because we really haven't done that. And then for functions, sort of modifies, packs, and then ships things out of the cell. Here is a diagram from under an electron microscope, and kind of a second one that's sort of drawn out showing what you're supposed to be seeing. But you see how it's a little short stack of sacs. Here's another diagram showing what I was just drawing for you, how a vesicle would break off, fuse with, and become part of the Golgi bodies. And then as stuff gets packed up and shipped out, little vesicles would break off. Notice this one now fusing with the cell membrane and then releasing the content. So it all works together as sort of one big unit. All right, lysosomes. You'll notice a little animation here in the corner while we're talking about lysosomes. Lysosomes digest things. So literally a cell might eat something. It would travel to the lysosome, fuse with it like a little vacuole of food, and then the lysosome would digest it. Just like our stomach digests food for us, lysosomes digest things for a cell. So it's an acidic environment. They're full of enzymes. If a lysosome was to break, it would kill the cell. Because if all the contents of a lysosome got released, it would basically digest the whole cell. So this is where that being compartmentalized is really important. Now, in our cells, where would we have lysosomes? A really good example of where we would have lysosomes in our cells would be like white blood cells, which do what we call phagocytosis. In other words, macrophages are certain white blood cells. That's their whole job, is that they float around and they look for things like bacteria, things that don't belong. They surround them and eat them, and then the lysosome is what would then digest those things. Technically, our cells do not need to be able to digest complex proteins, complex carbohydrates, because typically you eat your food, you digest it in your stomach. So by the time the nutrients get to your cells, they're pretty much digested. What's floating in your blood is not steak, it's amino acids, fatty acids, simple carbohydrates. You typically don't have starch floating around in your blood, you have glucose already floating around. But if we think of a single-celled organism like protists, pond organisms, they would need lysosomes because they're going to be eating their food whole. They don't have a stomach. So their cells, the lysosomes, would do the entire job of digesting. The other thing that your lysosomes will do is they will, what's called autophagy, which is basically recycling the cell's organelles. In other words, when an organelle gets worn out, mitochondria is old, it's not functioning anymore, the lysosome can break that back down into amino acids and carbohydrates and whatnot, and then you can rebuild it into whatever is needed by the cell. Excuse me. So lysosomes are basically sort of like a stomach for a cell. They digest things. And again, depending on the type of cell, like a white blood cell might have a lot of lysosomes, whereas a skin cell may not have very many, because there wouldn't be a need. So it really just depends on the job of that specific cell. So that's what they do. And this is just a diagram showing, again, lysosomes. They could fuse with a vacuole and then digest whatever was in it. Not really an obvious thing to identify from a drawing. It would probably be very unlikely that you would be asked to identify a lysosome in a picture, because they kind of just look like a little sac, just like a vesicle or a vacuole. There's nothing really obvious about them from a picture. Which brings us to vacuoles. So in animal cells, vacuoles, they don't do a lot of real exciting things. They're little sacs, like a vesicle, and they could carry food, for example, or store food. I would say from a drawing, the vacuoles that you would want to be able to identify would be this one, the central vacuole. In a plant cell, a vacuole is really, really important. In fact, it's actually bigger. If this was the nucleus, the vacuole is actually bigger than the nucleus. The vacuole's huge. It's one of the things that would identify a cell as very obviously being a plant cell. And vacuoles, the central vacuole in a plant cell, is very important to the entire life of the plant. Because what happens in a plant is that, remember how they take in water? And we saw this. You guys saw this in the water lab, how there's those little veins, those xylem that take up water. The water will leave that and go into the cells. What it'll do is it'll go to the vacuoles, and the vacuoles will swell up, sort of like think of a water balloon. And the more water you put, the more pressure. And this basically creates pressure, and this causes what's called turgidity, which is basically another word for stiffness. So the reason why the leaves of a plant are stiff and the stem doesn't hang over is because of the vacuoles inside every cell that are full of water that are pushing on the cell walls. So what happens when you don't water your plants? They get wilted. Why do they actually get wilted? They get wilted because as the water evaporates, now my vacuole, my little water balloon, it shrivels up, which means that my entire cell wall, this is basically what will happen, it'll collapse. And if that happens in all the cells, now the leaves are all wilted and the stem hangs over because there's no pressure anymore to hold them up. So the vacuole in a plant has a really, really important job that's kind of unique to plants. It's what gives the entire plant its stiffness. It's why the stem can stand up. It's why the leaves hold their shape. You don't water your plant and the water evaporates, the vacuoles shrivel, and the whole plant basically gets wilted because individual cells are collapsing. We're actually going to look at this in a lab in the next chapter. We're going to put salt water on some leaves, and you'll actually be able to see this on a microscopic level. You will be able to actually see them shrivel up. The other kind of vacuole that's kind of unique is what protists have. Remember, protists are the little single-celled eukaryotic organisms. They have something called a contractile vacuole, which actually can contract. The problem with a protist is that they're living in a lake, fresh water, and water tends to leak into them. So what will happen is, in theory, if water kept leaking in, a cell could explode. So what they have is called a contractile vacuole. It looks almost like a little flower. I have a picture of it on the next slide. And it will actually swell up. It'll suck up all the water, and then it just literally squirts it out, almost like a little bladder. Sucks up all the excess water and squirts it out of the protist. And that way they don't have to worry about exploding from absorbing too much water. And I'll show you a picture of it here. So this is animated at the bottom. It's not a great animation, but you see how it's squirting out water? It looks like a little flower. So it basically, in a protist, the contractile vacuole collects water as excess water accumulates inside, because this is living in a freshwater environment. It swells up, like you can see there, and it squirts out the water. Over here on the right side, you can see at the bottom a contractile vacuole that's full, and then one that's empty. But to me, it looks like a little flower. And then at the top, you see the central vacuole in a plant. This is actually, looks like an electron microscope maybe. But notice, it's the biggest thing there. It shoves everything else out of the way, and it's literally creating the pressure to hold that cell stiff, which then, if every cell is stiff, you now have a nice stiff leaf and stems and all of that sort of thing. So when those collapse, all the cell walls sort of cave in, and that's when a plant would look wilted. So that's contractile vacuoles and central vacuoles. All right, and that brings us to three organelles that have to do with energy. Two of them I'm sure you're very familiar with, the mitochondria and the chloroplast. So mitochondria are an energy-related organelle. They are the site of cell respiration, which, just a reminder, if you don't remember what that is, that's basically where they take glucose and oxygen, and they break it down into carbon dioxide and water, and in the process, they generate ATP. They capture the energy from glucose in ATP. And ATP, which we'll talk about in another chapter, is sort of the energy source for cell reactions. So that's mitochondria. I know everybody knows mitochondria because when I ask, everybody will always say, oh, it's the powerhouse of the cell. Like, that's been drilled into your head from, I don't know what, magic school bus, I'm not sure. But powerhouse of the cell, I always get that for mitochondria. The other energy-related organelle is the chloroplasts. Chloroplasts capture the sun's energy and do photosynthesis. So they basically take the sun's energy, kind of the opposite of this other reaction. They take carbon dioxide and water and the energy from sunlight, and they basically build glucose. They release oxygen as well. But so these are both energy related. The chloroplasts build up glucose, and then the mitochondria actually turn around and release the energy that was stored in glucose. So plants have both, so they can do both. They can capture and make glucose, store their energy, and then break it down as they need it. So that's why plants don't have to eat. We have to eat because we can't generate our own energy. We can't generate our glucose, so we have to take in our food sources, and then we can release the energy from them. So now, the third energy-related organelle most people don't really know about, and that's what are called peroxisomes. So peroxisomes do a lot of things, but the one thing that I would want you to know about them is that they break down fatty acids. So when you are, and then they basically give the broken down products to the mitochondria, which can then continue to generate energy from fat. So when you're on a low carb diet because you're trying to lose weight, your peroxisomes are doing a lot of work. Because you are, instead of burning carbs, you're burning fats. The peroxisomes are actually the ones in the cell that start that breakdown of the fats into simpler, they basically break big fatty acids into little pieces. That goes into the mitochondria, and then you can generate energy from it. If you think about peroxisome, you probably think it sounds kind of like peroxide. There's a reason why it sounds like peroxide. The product, they make a waste product. They make peroxide as a waste product, and then they have an enzyme in them called catalase that breaks the peroxide down. What happens when you put peroxide on a cut, besides that it burns? Anybody ever notice? Yeah, it like bubbles and fizzes. If you've never noticed, we'll do a lab where we're actually going to work with some peroxide. But when you put peroxide on a cut, it bubbles. The reason it bubbles is because of your peroxisomes. Technically, peroxide is not killing any bacteria. Just a little FYI, it's not killing anything. What it does do, though, is the peroxisomes have catalase, an enzyme. You put peroxide on a cut, it immediately breaks that peroxide into oxygen and water. And the bubbles you see are oxygen bubbles. That will, first of all, that'll bubble out any kind of dirt, bacteria, whatever. So a lot of times, like if you go to a doctor and you have like a deep cut and you've had it wrapped up and they open it up, they'll pour peroxide in there. Why? Not to kill anything, but it's going to bubble. It's going to get oxygen in there, and it's going to bubble out all the dead cells, any debris, bacteria, things like that. Then they'll follow up with either like iodine or an antibiotic or something that will kill the remaining bacteria. So peroxide's great for bubbling things out of cuts, but you actually make peroxide yourself as a waste product, and your peroxisomes break it down. So it's technically not killing bacteria. It's just bubbling out, like I said, bubbling out debris and things when you use it.